can still do that. Let's move on to Bree Woods. We did a PhD together at UTAS, so we know each other pretty well, and did go to sea. And she will talk about her new database called Myctobase.
Hello, everyone. Can you see that okay? What screen is it showing?
It's showing the title of the slide at the moment. Yeah, the deep sea. Yeah, oh, good.
Okay. So today will just be really short and sweet. I just wanted to take the opportunity to introduce you to Myctobase, which is a circumpolar database of mesopelagic fish. And this is a data collation project and one of the major outcomes of my PhD. So I'd just like to thank all of the data contributors that were involved and contributed to the project. I think my computer's a bit slow. Okay. So as many of you know, the deep pelagic community predominantly consists of fish, crustaceans, and cephalopods that inhabit the mesopelagic and bathypelagic depths. Mesopelagic fish are a central component of the community. They dominate global fish biomass estimates with estimates of up to 10 billion tonne. And they contribute to an estimated average of 16% of the total carbon flux out of surface waters during their diurnal vertical migration. However, deep sea ecosystems are underrepresented in databases of marine biodiversity. And this was nicely illustrated in this 2010 paper by Webb. So this shows that marine species occurrence records are dominated by those in shallow seas, despite making up just 10% of the ocean. And then we see this steep drop off in occurrence records for deeper levels of the water column. And these data gaps extend to areas of the Southern Ocean. So the advent of biodiversity informatics has seen the development of open access biodiversity data repositories, such as GBIF and OBIS. And this is really in the context of a global biological monitoring system. However, much of the data for mesopelagic fish that are currently archived in these repositories are for species occurrence only, and they're often lacking the abundance or biomass information, as well as the methodological metadata, so the net type or the depth of trawl, and this really limits the type of analyses that can be done. So there are decades of data from net sampling of the pelagic environment for localized regions of the Southern Ocean as part of different Antarctic research programs. And Myctobase is really a first step toward integrating and standardizing the format of this data, so published and unpublished survey data, and specifically data on abundance, biomass, biodiversity, and the methodological metadata. So our specific objectives for this project are to provide a single findable, standardized, and citable resource for multiple combined data sets. Also to provide the baseline data upon which to measure the status and future trends of the mesopelagic fish assemblage, and to support further research such as investigating the patterns of biophysical drivers or diversity. So I'm not going to go into the specifics of how the data were processed or standardized because it's quite boring, and you can just read about that in our data paper, which I can have sent around later. But basically, the data are structured as you see here. So an event, which is the net haul, that contains the information on the gear type, the date and the time of the trawl and the duration, sampling protocol, depth sampled and volume filtered. And then within each of these net hauls, we have a group occurrence. So within the group occurrence, we've got species abundance and biomass data, so they're grouped by the lowest taxonomic level possible. So many don't have species information, so we've just gone the next level up. And then within the group occurrence, we have an individual occurrence, which are individual records of the sex, the maturity, the length, and the weight of the individual. Now, to show you the spatial coverage, so to date, Myctobase currently holds about 4,700 net hauls from 3,775 sampling stations and 72 different research cruises from across the Southern Ocean. As you can see, the highest concentration of data points are within the Atlantic, and we have this big gap in the Pacific. And vertically, the data extend from the surface down to a maximum depth of 2,000 meters, with more than half of the trawls currently taking place in the epipelagic. And looking now to the temporal coverage within the data set. So as you can see on the X, we've got time. On the Y, we've got, on the Y, we've got the number of trawls. On the X, we've got time. And so the data span from 1991 to 2019. And as expected, most of the data from these summer months. So about 76% of the net hauls within Myctobase are for summer. And this is just showing the 4 most abundant fish families in Myctobase. And as you can see, myctophids make up the highest number of records within Myctobase, which is why it's called Myctobase. In particular, the highest number of records for the Indian sector in the subantarctic for myctophids. So you can access the data in a couple of different ways. So it's currently up on Zenodo, GBIF and OBIS. You can also access the data using R by if you install and use the Blue Ant package, which I thank and acknowledge Ben Raymond for setting that up. So I can have this slide sent around, or just the code or the link below if you're interested in doing that. And we will continue to update the database as we get more data. So if you do intend on using the data in any way, I suggest you take a look at the data descriptor that accompanies the data. This is published in Scientific Data and contains specific information on the data collection and processing, the data records, technical validation, and the usage notes. And we really welcome further data input to the database. As you saw, we had a massive gap in that Pacific region. And we hope that the database will continue to grow and become the platform for maintaining this type of data. And the other outcome from this work is we currently have a habitat modelling paper in review with Frontiers. So I would be excited to share some of the findings from that at a future seminar. Thanks so much for listening. I'm really excited to see what people do with the data, and please don't hesitate to reach out to myself or Anton if you have any questions or if you would like to contribute data. Thank you very much.
Awesome. Thank you so much. That was pretty amazing. So nice to see it, finally published, and, you talking about this for years, basically. So, so nice to see it out and it's there, and, the data.
Oh my God, I know.
It has been such a big effort, because, how do you get the data? Did you just write to people and say, oh, do you have data? Or,
Well, I actually got to go overseas
yeah,
a couple of times to visit collaborators, which is really nice.
Yeah,
before COVID.
Yeah, fair enough.
Yeah.
But I mean, you have to know the right people to talk to, right? Otherwise, it's, their data is sitting in some drawers, or there's, otherwise it's kind of hard to,
Anton was a bit of a link between who the right people were for collecting data and whatnot. So
yeah.
Yes.
So what if, for example, I know some people now in New Zealand, and I think they have some mesopelagic fish data for the Ross Sea, so Pacific sector. Should I just contact you and Anton saying, oh, we have this data, do you want to edit it, or how does it work?
Yeah, so you can just send myself or Anton. Actually, if you send both of us an email, so
yeah, it makes
just
sense.
letting us know that you have data, and then we will send you a bit of a template.
Yeah, okay.
To put the data into, and then we can do the rest to input it into GBIF and OBIS.
Yeah, that's awesome.
And then in the R package, so anytime someone uses Blue Ant package to get the Myctobase data, it will be the most up-to-date version of that data set.
Awesome.
Yeah,
that's pretty great. Yeah, that sounds good, because I was wondering about the format it has to be in, but if you send a template, that's super easy then.
Yeah, so the template will guide you with the format that it has to be in.
Yeah,
and then we'll do like
makes sense.
Processing at our end. Yeah,
nice. There's some questions. George also asking, are there clear protocols for submitting data? I know researchers with jars of trawled Myctobase, we all do know them. We are them actually sometimes. Do they have to measure, or do they have to measure, or do you still have funding to come and visit?
Don't have funding.
Good. People have to measure then, I guess, and have a look. So do they always need size and abundance and biomass, or can it be only one of them?
I mean, yeah, like I don't have size and weight information for every single observation, but the data was there, so we thought, why not include it?
Sure. Yeah.
People will use it.
Yeah.
So whatever form the data is in is fine. We will add it.
Yeah, awesome.
Yeah.
Nice. Lauren is asking, any plans for this database to expand to other ocean regions outside the Antarctica? This is awesome.
I have been thinking a bit about that. That would be really cool. But yeah, as I said, I don't have funding, and maybe that'll be something that I can kind of apply for a postdoc and follow and keep following that. But yeah, maybe watch this space.
Yeah, if people have some funding, please pay Bree to do it. Yeah, and Bowen is asking a similar question. This is mostly in polar areas. Yeah, fair enough. Yeah, I mean, similar.
Yeah.
Awesome. How much different are the circumpolar mesopelagic fish from the tropical, subtropical mesopelagic fish?
Well, I've only ever studied mesopelagic fish in the circumpolar region, so unfortunately I can't really answer that question, but maybe someone else on here knows more about the subtropical mesopelagic fish.
You always feel like when people put their mesopelagic trawl things online on Twitter, then things look similar, but I can't even say. I guess there are species there are, globally distributed, and it's fine, but others might not. So I can't even say anything. It's like outside my expertise. I don't know.
The expertise is this narrow.
I know it's it's fine. Other things. Cool. Any more questions, people? Looks pretty good. Yeah, no, I think it's a pretty nice project, and thanks. I think let's take the opportunity to thank Bree and Michaela to give some amazing talks, because it has been pretty. I learned a lot, so I really enjoyed this call. Thank you so much for volunteering, letting me volunteer you to give this talk. Awesome, people. So the next JETZON call will be, I think, end of September or early October, led by Adrian again. So will be more UK / US East Coast friendly times. And then hopefully, yeah, then I'm in the field. So we will see how that call will be in November. But yeah, we will talk about that then. In the meantime, if you want to check out JETZON, actually I can quickly give you the website. I think it's that. Yes, it is. It's it's our website. So, and they're just refreshed it a bit. So if you want to check it out, there's some other projects. I see Bree just posted a link to the paper to. I can also send it around to all people that participated today, actually. So yeah, if you want to check it out and don't find the time now to download it, it's fine. And thanks everybody who joined us today. I will just stop recording right now.